So I'm hoping to leverage our understanding of what we've done so far and a detailed discussion of mitosis. Um, and so meiosis has two divisions. We've talked about that before. So there's meiosis 1 and meiosis 2. The key thing that happens in meiosis 1, this is where you have the chromosome number is reduced by half. So we start out with a diploid cell, and this is where the first time we have a haploid cell. It's going to divide again, these two divisions. And so then we end up with meiosis 1 or 2 going on. And this is a lot like mitosis. So there's some key things that happen in here. So ultimately, we're going to make 4 daughter cells from a parent cell. They're going to have half the genetic information of the parent. More about crossing over in the later slides here. And really, the purpose of this is to make gametes or sex cells for sexual reproduction. And they are haploid. They have half the number of chromosomes in them. So a detailed discussion of meiosis, I'm really going to focus on how it's different. So prophase, things coil and become compact. The homologous chromosomes are going to line up. And so if we have metaphase 1, so this is the first time they line up, this is where we have tetrads or homologous pairs lining up. And so you'd have 2 chromosomes lining up. When these come apart, the entire double chromosome is going to go to the 2 new cells. This is where the cells are now haploid. And so anaphase 1, when they separate, this is the key part in meiosis 1. And here's the picture of them. There you can see them lined up along in a double row in metaphase 1. In meiosis 2, there's no more chromosome duplication, but then it works a lot like mitosis. So in this one, in metaphase 2, this is where you have just a double chromosome lining up, and they're going to separate at their centromeres for one chromatid to go to each of the 2 new cells that it's forming. So it's no further reduction. It was haploid to start, and it stays haploid at the end. And so this is where you have the sister chromatids separate. And like I said, that's a lot like mitosis. So here's a picture of that going on, and you end up with 4 haploid daughter cells being formed. If you were to compare and contrast these, I tried to put this slide in the last video or the last PowerPoint, but also in this one. They are the same in a lot of ways. We have both, they duplicate their chromosome once in interphase. In mitosis, we've got 1 round of cell division. In meiosis, we've got 2. How the chromosomes line up in metaphase, they're single file in mitosis, and they line up as tetrads in meiosis. We're going to make 2 daughter cells that are the same as the parents, or we're going to make 4 daughter cells that are half the same as the parent cells. The purpose of these is for growth and repair or reproduction, asexual reproduction of a single cell organism, or it's to make gametes or sex cells for a sexually reproducing organism. These are somatic cells as opposed to gametes, and we've got diploid cells produced here and haploid cells produced down here. They are both cell division, they both produce new cells, and that's a pretty good summary. So hopefully all those points make sense, and you can put that together. So the key thing about sexual reproduction is that it produces a much greater amount of variation than asexual reproduction. And so sexual reproduction is key when we talk about evolution. So the variation is basically the raw materials for evolution. Now, if you don't have sexual reproduction, then you're relying mostly on mutations to provide that variation. But the idea that sexually producing organisms have a constant recombination of genetic traits or alleles is really important. So the frequency of alleles, the frequency of those traits can change as a population evolves. And so not necessarily, the express traits, but even the alleles that are coded in the population. So dominant traits are not always more common than recessive traits. As an example here, if you have a straight hairline, that's a recessive trait, but it's far more common than the dominant trait of the widow's peak. And so if you have a hairline that has a little little point on your forehead, that's a widow's peak, and that's a dominant trait. But there's far more straight hairlines out there in the world than there are widow's peaks. The other thing is mutations and alternation of genetics, an alteration, sorry, of genetics can increase genetic variation and leads to changes in a population. So this is the raw material for evolution. And some of what we know about meiosis can also contribute to that variation. So we're going to talk more about meiosis and the inherent things that happen in meiosis that really contribute to all that variation that we see in sexually reproducing organisms, so plants or animals. The reasons for sexual reproduction, the variation in sexual reproduction, is that when those tetrads line up, so the tetrads are going to line up, those homologous chromosomes, remember you have one maternal and one paternal, you're not going to have all the maternal on one side and all the paternal on the other side, right? You might have a mixed match. And remember, you have 23 chromosomes, 23 pairs of chromosomes, we should say, that are all lined up this way. And so the idea that they're all lined up and are going to be sorted equally is not the same. So we've got 23 combinations and permutations, and this is going to make a huge number of possible gametes formed. If you take 2 to the 23, this is what the math would be. You end up with over 8 million possible different egg cells and over 8 million possible different sex cells, sperm cells that are made in humans. And so the likelihood of making the same egg cell twice is almost impossible. And so this is going to make a lot of variation. This is called independent orientation. We're talking about the alignment of the maternal and paternal chromosomes facing a given pole. So in this one, you've got a huge amount of variation. Now, the next part of this is that we know that these sex cells have to come together. So that's what fertilization is talking about. Now, it's not random. You shouldn't randomly fertilize sex cells. But the idea is that we can't control which two sex cells of these 8 million are coming together. So this further increases the possibilities of the different offspring that would be made by two of the same parents. And so as an image here, this is just showing the different options that happen with independent orientation. And your printouts might not be printed in color, so that's kind of everything on these slides. Otherwise, they look exactly the same. So this is showing independent orientation. And then from here, you would have random fertilization. And again, in this, you can't control which sex cells are coming together. So all of this creates a lot of genetic variation. A third cause of variation is when we have those homologous chromosomes lined up beside each other. So they're lined up nice and neat beside each other. They might actually be so tight together that they actually get sort of tangled up, if you will. And so I usually do this in the class standing next to somebody, and we put our arms up beside each other and cross them over at the elbows. So this crossing over is basically them getting tangled up. If they cross over, if this is our elbow, the fancy name for that is a chiasma. And we're going to basically exchange a certain amount of chromosomes. A certain amount of genetic material. And so when we do this in the class, I talk about I would walk away with the other person's forearm and they would walk away with my forearm. Hopefully you're imagining that. So these non-sister chromatids join at the elbow, if you will, the chiasma, and they cross over. They have an exchange of genetic material, and that's what crossing over. So now you don't have a complete maternal and paternal. You have more combinations that are possible to go into those gametes, and this is more genetic variability in sexually reproducing organisms. So understanding the process of meiosis can help understand genetic differences. And so a common, well, not common, but it can happen, process that happens in meiosis is non-disjunction. So non-disjunction, it can happen in two ways. So non-disjunction could be that the homologous chromosomes are having such a good time together that actually both of them go into the new cell, right? So you get extra chromosomes in one cell and a shortage in the other. Or you could have a chromosome in meiosis 2, and instead of it separating as at the centromere, both sister chromatids are going to go into one of the gametes. And so either one of these, you end up with an abnormal number of chromosomes. Usually you get an extra number. A common example or a familiar example would be Down syndrome. So the autosome number 21, you would actually have 3 chromosomes in that location when you looked at a karyotype of an individual. And so you end up with an extra chromosome in each of the somatic cells. And the key thing here is that if an error or a difference is made in meiosis, that's going to lead to an offspring with genetic differences in all of their somatic cells. So this is an image of what I was trying to illustrate. So here you can see how both of the tetrad, both of the homologous pairs are going into one of the cells, leaving a shortage. And then here you see they're not separating at their centromere. And so we end up with gametes formed that have extra chromosomes and gametes that are formed that have a shortage of chromosomes. A lack of chromosomes is often a more significant difference in offspring. So we don't see that is common. You can have extra information, and that's a more common thing. So other things that can happen in meiosis that can cause to birth defects in in if it's in meiosis or if it's in a somatic cell, they could lead to cancer. You could have a deletion. You could have a loss of a chromosome section. You could have a duplication. So maybe if I was crossing over with a homologous chromosome, maybe the homologous chromosome walked away with both of our forearms, right? That would be a duplication. I would walk away without a forearm. That would be a deletion. I'd be missing information. You could walk away, so the one chromosome could walk away with my forearm on upside down. So it's a reversal of a chromosome segment, and that's going to read the information differently. It's going to change the recipe. It's going to change the coding and the genetic information. You could also have a forearm put maybe attached to somebody's knee or somebody's foot. So it's in a different location on the non-homologous chromosome. So these are all ideas of errors or differences or mistakes that can happen in meiosis, and it's a big effect when it creates gametes, but it can also have an effect if these are happening in mitosis and making somatic cells. So this is just an image of those. And again, we think of mutations as bad, but I would like to think more that these mutations are the materials we need for evolution to happen. They are the raw materials. They are the differences that can maybe give an advantage and change the gene pool in an organism.